It works. All right, that's the first hurdle. Most of you I know, some of you I don't, and we can talk after this. Some of you have been with me for decades and have gone a long way down the path, have DNA testing and everything. Others I met is, in one case, yesterday for the first time here, and DNA testing has just been discussed, and we're in the process of ordering it, and everywhere in between. But this is a difficult lecture because everybody is at a different part in the journey. Mitochondria are the parts of the cells that make the energy. I mean, everyone hears that. But energy is life. Every cell needs energy for almost everything that it does. So when you see the list of things that mitochondrial disease makes, what it can be related to, it sounds like everything. And to some degree that's true, because if the cell doesn't have enough energy, it can't pretty much do anything. So when you have a problem in bioenergetics or in making energy for a cell, you are predisposed towards a lot of different disease manifestations, and every patient is very different. So every patient is different, and you're all in different parts of your journey. Some of your kids are very, very mildly affected, others are very severely affected, most are somewhere in between along the entire spectrum. I've also been told, including by my wife about an hour ago, that I cover too much area, my slides are too complicated, and I talk too fast. That's all true, and that will happen again today. It is complicated. I'm looking at this lecture more to tell you what the possibilities are, because we're in the future now. We're always looking to get ahead, and I hope 10 years from now I can say that again, and these are the dark ages. But what we can do today is far beyond what I had dreamed 10 and 15 years ago that we would be at. It's really the difference in the technology and the knowledge that we can do today is really astounding, and not in every single case, but most of the patients are doing much better. Mitochondrial disease used to be considered progressive and fatal. There have been exceptions, but I think I've had 2 patients die in the last 5 to 7 years or so. I have hundreds of complicated patients. There's been a lot of changes, and it's a really good time. So what I'm going to be going over is I'm going to be concentrating on genetic testing, because that's the biggest single issue. But I'm going to be covering a lot of little things here and there, and I'll just talk about it and I'll go off to the other topics. So wait a minute, you could have spent an hour on that, you spent a minute at most. Yeah, I'll do that a few times on a few topics. But at least you'll know about it, and I'll try to get you to the right resources as to you can look up more, you can ask later. Regarding questions, that was the big issue when you have a group like this. A lot of questions are personal. This is being taped, and it will be available on the website forever. So if you talk during the presentation, your voice will be forever on the website, and your question. If you hold the questions till the end, we will turn off the recording, and we will tell you when we turn off the recording. And then if you have questions that you think that are general questions that others might learn from and that are not too personal, we can have that in this situation, so we can cover some of the larger areas. For personal questions that you'd rather have it just with me, I mean, I'll be circulating afterwards, and we can do that. So, okay, with that in mind, start the presentation. What this is about more than anything is connecting the genotype, which is the genetic information, to the phenotype, which is the clinical manifestations in the patient. So here is the DNA sequence, here, and then we're connecting it to disease. And in mitochondrial disease can be anything from cyclic vomiting to autism to actually much more severe multi-system problems. And Kylie here is one of our models. Showing the bear that my daughter Caroline over there gave to her. That was many years ago. You're a teenager now, so sorry to show you stuffed animal from the past. So, okay. And this actually is the mitochondrial DNA sequence of the control region, in case anyone ever wanted to know what that was. It's a real sequence. Okay. The first patient here is like a milestone to me, Ryan. I saw Ryan on Thursday again. And he was my last patient with cyclic vomiting to put under control. I mean, I've seen some patients as recently as yesterday, and of course they're not in control yet, but I'm talking about ones that I've been following for a year. Sometimes it takes a while, but of the patients I've been following for at least, I think, 9 months, nobody vomits anymore. Maybe if they have a stomach flu or something really bad happens, they may break through and have an episode, like one a year or so. But everyone's under control, and that's a huge difference from where it used to be that patients used to go into the hospital every month or so. It shows, and a lot of that is a story of the technology and the knowledge that we have. Let's see. I got permission from the family to use his real name, and I haven't gotten the photo yet, so I haven't changed the Randall to Ryan yet. So it's the same one. And he presented as a teenager with episodes of vomiting. Many of you have cyclic vomiting because that's kind of my area. I'm the Cyclic Vomiting Syndrome Association. My official title is the Vizir of Vomit. I wanted Emperor of Emesis, but that was taken. That was the coolest title. But anyway, there's a Prince of Puke too, but you know. So anyway, I've been involved in that for about almost 20 years now. And Ryan, I can say his name, I saw him yesterday, Thursday. He had episodes of cyclic vomiting. Some of you live that, some of you have never heard about it before. It's where you have nausea, vomiting, and lethargy or tiredness. And they come in distinct episodes. You go into the episode, you're really nauseating, you have vomiting, you're really, really tired. It lasts for hours to days, and then it goes away until the next episode. The average is to have episodes every month, but it varies tremendously. Some people have it at random. Some people have it every 2 weeks, sometimes every 2 months. Some people have like 3 or 4 a year. It may be a clockwork, and it may be associated with stress, or it just may come at random and nobody knows why. But in Ryan's situation, he had it exactly. I don't know why the font is so small in here. Anyway, that's really weird. It's supposed to be larger. 88 to 92 days. It would almost always be 90 days. Occasionally it'd be 89, sometimes 88, sometimes 91, sometimes 92, but almost completely at 90 days. So the family was able to schedule all their trips and vacations and everything because they knew exactly what day he was going to do it. Maybe a day before, maybe a day after, at most 2 days differently. Imagine how the healthcare system looked at this mom. Yeah. He's going to have an episode in 37 days exactly. Yeah, I mean, you can imagine how they took that. And he does, I mean, everyone I've ever seen with cyclic vomiting syndrome has a degree of mitochondrial dysfunction. Many of them have straight mitochondrial disorders. In Ryan's situation, he had ketosis during episodes, which showed that he had mitochondrial dysfunction. He had some other things, which you cannot read because the slide changed. It's fine. Chronic fatigue, some cognitive delay, autism, GERD, constipation, choking episodes, obstructive sleep apnea, some other things, not just cyclic vomiting, which is the usual, but not in all cases. And he failed treatment with everything. Everything that is known to be done, he was on everything, and he didn't get any better. And we did genetic sequences on him, and found a variant of interest in the sodium channel 4A. It's, you'll be hearing a lot about ion channels today. Ion channels are proteins that cross the membrane of the cell that allow the ion that with sodium channel allows sodium through. Sodium is salt. And when salt goes through the membrane, the nerve cell fires or sends a message, if you will, and an impulse. And that's because it's electrical, because sodium is charged. And to have nerves function, you have to open up ion channels and let the salt through, and that's how nerves fire. And what happens is when channels are leaky, you get things like migraine and cyclic vomiting and chronic fatigue and irritable bowel and all the stuff that we'll talk about. And so he also has another variant in a gene which is involved in making protein, which probably has something to do with it as well. I put him on acetazolamide, also known as Diamox, which is a diuretic, and that's used to treat the sodium 4A channel, and his episodes went away completely. He's now at day 476. 2 days ago, Mom said it's day 474. He hasn't had an episode since then. So he was the last patient to get under control. And so you say, well, okay, channelopathies. Isn't this a mitochondrial talk? The mitochondrial talk is next door. This is a channelopathy talk. I'm sorry if you're in the wrong place. No, the channels, the nerves function because channels open at the right time. If channels are leaking, the pumps have to work all the time to take that sodium and put it back where it goes. So mitochondrial dysfunction can be because you can't make enough energy or you use it too quickly. Actually, I find more patients have mitochondrial dysfunction because they use energy too quickly than I find patients have it because they can't make enough. Many patients have both. This is an example of mitochondrial dysfunction, not because the supply is low, but because the demand is too high. There's a leak of the sodium current, and the pumps have to go 24/7, 365. That's using energy, so the patient runs out of energy and has mitochondrial dysfunction because of the demand side. Okay, so having gone over that, who am I? Many of you know who I am. I'm many different people. I'm a clinician foremost. I'm an MD. I see patients. The two areas that I do mostly are the weird migraine stuff, including cyclic vomiting syndrome, hemiplegic migraine, etc., and autism and other neurodevelopmental disorders. And we'll get to those. But I see patients in all sorts of things, a lot of chronic fatigue, chronic pain, anything that's common in mitochondrial dysfunction, I do that. I'm also the Chief Medical Scientific Officer of NeuroNeeds, the company that makes that. That's my mito cocktail I've been using in some way for 25 years now. Finally put it in a jar, and we can talk about that later. I have been the medical director of multiple DNA sequencing companies, 4 different ones in the past, including Cortigen for 5 years, Lineagen, and some others that no one's ever heard of. And in the past, I do not work for any company right now. I'm a free agent. I negotiate prices with labs all over the world to get the best product at the lowest price. I'm also an expert in legal cases. I take on a lot of legal cases, particularly lately it's been mostly medical child abuse, Munchausen by proxy. That's been, unfortunately, a major change over the last few years that those cases that used to be around have now exploded. And that in the past I've been a researcher. I'm actually writing 2 papers right now still, but I don't do nearly as much as I used to before when I was a faculty at USC. Okay, next patient to go over is Payton. I think she's about 18 now. She was 14 when we figured out the diagnosis. She did have cyclic vomiting from her first birthday until about age 10. As often happens in cyclic vomiting, but certainly not always, it then morphed into migraine. It was migraine 24/7, 365. She had chronic pain not just in the head, but throughout her body. She had multiple admissions for bowel cleanout. She was a student, but the main problem was chronic fatigue. Her chronic fatigue was so bad that as she came downstairs for breakfast, that she was wiped out the rest of the day and couldn't do anything more. So she was a good student on her laptop in her bed, but she was completely disabled by this disease. How did that happen? Well, we did the sequencing on her, didn't find anything rare. Found one variant in the TRAP1 gene, which is, here's the variant, P for protein, isoleucine changes to valine at amino acid 253. So now you can read these things. It's a common variant, and 1% of people from Northern Europe have that variant, about 0.5% from Southern Europe and Latinos. So, okay, 1% of the population, there's millions of variants that are in 1% of the population. Why did I think this one was a problem? I thought this one was a problem because the gene explained her issue, and I at the same time had 2 patients very similar to her that had exactly the same variant in that gene. So I started to look at patients and found about 30 patients with the same variant, and they all had the same issues. They all had pain, fatigue, and GI problems. And so we published this as TRAP1-related disease. And here's a little cartoon of what TRAP1 looks like. 2 subunits. 2 proteins are identical. The one in silver and the one in pink are 2 subunits of this protein. It acts like a crab. What it does is it's a chaperone. You know what chaperones are? Chaperones take things where they're supposed to go and they don't do what they're not supposed to do on the way there. That's a chaperone. A molecular chaperone takes the molecules where they're supposed to go and they don't unravel, which is what they're not supposed to do, on the way that they're supposed to do it. The mitochondria is a place that has, it's a very hot location. That's where metabolism happens, that's where heat is made, a lot of free radicals and other things. The furnace burns very hot in mammals, and there's a lot of things in the mitochondria that can damage molecules. So what the chaperone does is it's like a claw. It grabs onto proteins, holds them into place so they can't unravel, takes them where they're supposed to go, and then lets them go. So that's how the chaperone works. And TRAP1 is the only really molecular chaperone for the mitochondria. And it uses ATP to do that, to be able to grab onto proteins. You see there's a little green here you probably can't see. There's 2 molecules of ATP in here. It breaks the ATP using energy to do that. Now, in the patients, all had mutations in the amino acids which touched ATP. I think what's happening is that it's an ATP eating machine. I haven't proven that yet. Is that it's using up the ATP without doing anything, and so that the chaperone's not protecting the proteins and it's using up energy too quickly. So, and this particular, again, 1% of the population has it. I have a large number of patients that have this variant. Again, chronic pain, fatigue, and GI dysmotility. So what we did is, with colleagues at Georgia Tech, is they took their computer system over the entire weekend, the whole Georgia Tech computer system, and ran the mutant TRAP1 molecule with the mutation in place, and the wild type, or the normal TRAP1 molecule without the mutation. They looked a tiny bit different in three-dimensional space on a computer simulation. We know exactly what the TRAP1 molecule looks like because people have done X-ray diffraction and gotten a picture of it. So if they know what it looks like, they say, how would the mutation change the way that it looks, and come out what they think that it does. Then you take every single chemical that could possibly ever exist, and on a computer decide which will bind to the mutant and not bind to the wild type. Because what I wanted to do is I wanted to jam something in the mutant binding site here to destroy the mutant molecule. And according to the computer, comes up with SAMI. So we tried SAMI in a couple of patients and they got a lot worse. Stopped the SAMI and within hours they got better. Okay, so SAMI probably doesn't jam the mutant, it probably super energizes the mutant. So that's not working. The next one we tried was Kytril. Kytril is a prescription medication. It's used for chemotherapy-induced nausea and vomiting. I've used it as cyclic vomiting before because it's a great drug for vomiting. It's just extremely expensive. So they only use it in cancer patients. We tried Kytril and it worked tremendously. Her fatigue melted away completely. She went back to school full time, then she went to half-time for a while. Her chronic pain went away. Her bowels no longer have cleanouts anymore. She's almost entirely different patient. And that's based upon doing the molecular and then taking the next step forward, connecting it with the other patients, and then going the next step forward to see what works and what doesn't work. We say, well, I'm interested in, cyclic vomiting and autism are kind of my 2 little areas that I see a lot of patients on, and people think, well, they're very different. But I got interested in autism mostly because autism was in a lot of the brothers of the sisters with cyclic vomiting syndrome. I saw that over and over and over again. And the same genetics that will predispose towards autism apparently predisposes towards cyclic vomiting syndrome. Autism can be in boys and girls. Cyclic vomiting can be in boys and girls, but it tends to be cyclic vomiting is a little bit more common in girls, and autism is a lot more common in boys. So a lot of the patients that had the 253 change in the TRAP1, a lot of their brothers had autism. So I said, well, maybe that's contributing to the autism. So I chose Carter as my first patient. Carter has the 253 mutation, and he had very severe autism. If you go to a restaurant, he would run around the floor and everything, and you would not sit and eat. They couldn't go anywhere with him. One of the parents had to be home with him at all times, so they couldn't take him anywhere. So we put him on Kytril, and these are the things that improved according to the developmental specialists. And then the ones that have the asterisks on there got better when he was on Kytril, and then got worse when he was off of Kytril, and got better again when he was on Kytril. Unfortunately, we got a lot of times to see how he did off of Kytril because Kytril is a very expensive drug, and the insurance company kept denying it, and they kept running out. the pharmacies. So he went off of Kytril 4 times that I know of, and he would get worse each time. He'd go back on and he'd get better. So this is not just a placebo effect. The school was noticing it, the OT, the parents, and it was on and off. So it's to show, again, that what can be done if you know the molecular problems. Okay. So the FDA requires that I say this. Kytril is not approved by the FDA for the treatment of cyclic vomiting syndrome or autism. And this is molecular medicine. It's called personalized medicine, where you find out exactly what's wrong in the DNA of a single patient and then use a drug to try to fix that patient. Obviously, this is not a situation where you've had hundreds of thousands of people through a clinical study and that you've proven that the drug works and that it's safe. When you have one patient that you find out what's wrong with their DNA, it's personalized medicine and it's not FDA approved, and quite frankly, it's very difficult to find doctors that are willing to treat under these circumstances. Okay, here we go. Okay, Carl, who's not his real name, that will become apparent why, had episodes of abdominal pain, nausea, vomiting, and he became very pale. The abdominal pain was by far the worst aspect. So by the nomenclature of medicine, it's called abdominal migraine. Abdominal migraine and cyclic vomiting syndrome, in my opinion, are exactly the same. If vomiting or nausea is your problem, your major problem, it's called cyclic vomiting syndrome, but many of them have abdominal pain. If abdominal pain is your major problem, it's called abdominal migraine, but many of them have nausea and vomiting. So anyway, you could say that he is an abdominal migraine patient. If you're under your 18th birthday and you come in with an illness, and you have this illness over and over again, and nobody can figure out what's wrong with it, what do they think is wrong? They think the mother poisoned the child, or the mother's making it up, or something like that, right? But after your 18th birthday, the kid then becomes an adult and is now a drug user. So it is the way that it is, and I'm totally against it, but it's the system that we are in. So he got labeled as a drug addict, and through a major metropolitan area, not Los Angeles, he ran out of emergency rooms. One of the 10 largest cities in the country, he ran out of ERs. He was labeled drug addict everywhere. And they went to another metropolitan area and it started getting labeled there too. We did the molecular on him and we found 3 known mutations in the RY. Oh, first of all is that we had him under control with the cocktail and amitriptyline, and then he went off of it when he got better, which a lot of people do, and then he relapsed and the cocktail and the amitriptyline wouldn't work anymore. Sometimes it happens. So then we did the molecular technology and found 3 known mutations in the RYR2 gene. This is a calcium channel, not a sodium channel. And based upon this, I put him on high-dose propranolol and the disease went away. He's also gone off and on propranolol several times as he's moved from state to state looking for a job and going with family. And at the same time, insurance has been denying it, and also his compliance has not been perfect. So when he goes on propranolol at high dose, his disease gets better and it goes away. And when he goes off of it, he ends up back in the emergency room with severe pain again. And he's been doing this for a few years now. This is a really good model of psychosomatic disease. It has a bad name, but let me explain it a little bit. The old way of thinking, the old Freudian way of thinking is that you're really emotional and that you will then manifest things like somatization. You will feel things that are not real because of your emotionality, because you can't handle something or there's something like that. And unfortunately, a very large number of physicians and other people still believe in that Freudian. Well, I almost said something. Anyway, for 150 years ago, he was really a pioneer, but it doesn't quite work today. You can do a little better than that now. So what happens with the RYR2 is a calcium channel across the endoplasmic reticulum. So calcium is pumped into the endoplasmic reticulum. an energy requiring process, and it's there for the cell is triggered. When the cell is triggered, the calcium is then released through the RYR2 calcium channel, and the calcium does what it does. If it's a heart cell, it says it's a heart cell to beat, to contract. If it's a muscle cell, it contracts. If it's a gland cell, it secretes. If it's a nerve cell, it fires. Whatever that cell is, the calcium is the message to do your thing. So the RYR2 is really the trigger that makes the cell do what it does. Now RYR2 is opened up at norepinephrine. Norepinephrine is the stress hormone. So the way that it works is that, okay, so you're a caveman, you're out in caveman society, and you see a tiger. You realize that the tiger is likely to eat you, and so you get stressed. Your brain makes norepinephrine. The norepinephrine turns on your heart and your muscles and your nerves to energize your body and to give you the energy and the the clarity of mind to be able to fight the tiger or to flight from the tiger. It's the fight or flight response. It's the stress response. It also turns off the skin and the kidneys and the digestive system and the liver and everything else, because everything goes to the brain, the heart, the muscles. That's where you need everything. Now, in modern day life, we just don't have tigers every year or so. We have little tiny things that bother us all the time. Stress is constantly norepinephrine is constantly being released in our busy lives all the time, usually in smaller amounts. What if your RYR2 gene had a mutation in it that it opened up the calcium channel with just a little bit of norepinephrine, full-fledged, just opened up completely like there's a tiger about to devour your babies? That's what these patients have. Every tiny little thing that happens, their calcium channels open up and they go into a fight or flight response. So what's that do to the body when they're in a flight or flight response all the time? Well, first of all, their mitochondria are turned on all the time, because that's obviously in a fight or flight response, you want the mitochondria on full steam. Everything gets opened up. So their mitochondria burn out basically because they're on all the time. They also get a lot of GI diseases and a lot of other things by systems that are turned off. So when you have these mutations, you get a lot of things like cyclic vomiting and other GI problems because their GI systems are turned off, but at the same time they have mitochondrial dysfunction because their mitochondria are normal, but they're turned on constantly, so they're dysfunctional. So what we do is use propranolol. Propranolol blocks norepinephrine from opening up the calcium channel. So it's actually a specific therapy which blocks the stress response. The other thing is that we do is we use a lot of antioxidants, because no matter how much propranolol, some of it's going to slip by, the mitochondria are going to be on too much, and we need the antioxidants. Over normal mitochondria that are on all the time produce the same free radicals that abnormal mitochondria make. So they need antioxidants. There's also, you can see, a psychiatric or a psychological aspect of the disease. To say that you have to be like a monk sitting on a mountaintop and you have to say your word and just not let anything get you, because it's not worth getting into the disease. It's not that you have a psychiatric problem. It's not that your mind can't handle the stress. It's that every tiny little stressor causes your body to tear itself apart. So you just got to try to control the stress. So those together actually work very well. This is a tiny little cartoon. It just shows that the membrane of the nerve has a lot of channels. It actually has several hundred channels that allow sodium and calcium and potassium and everything to go through. And then, of course, there's pumps as well that pump it back to where it's supposed to go. If the channels are leaky, the pumps have to go all the time. Okay, next patient is Margo. Her cyclic vomiting was mostly dizziness, but there was some vomiting and nausea as well. And her dizziness she was under reasonable control. Then when she got to be 12, her dizziness got so bad that she couldn't walk. It made her totally disabled because she couldn't go anywhere. She couldn't go to school. She couldn't be out with her friends. She was at home all the time and having to learn on a computer. And did the DNA sequencing and found a variant in the same RYR2 gene. Gave her propranolol and antioxidants, and the disease melted away completely in her. So Margo became a teenager, and she volunteered for Children's Hospital. She had a full-time job. She was taking full classes and AP classes, and just kind of overdid it because for years she was like a hermit in her house. She's actually now in college. Okay. My daughter's a little bit older now. This is just to show that, when you have an advanced technology like this, it can seem like magic. But I want you to understand how the process actually works. Because the process involves me and the laboratory and everything, but it also involves you to a large extent as well. And the way that it works is Payton right here. His case is going to show you how the system works. So he lives on the East Coast, and he has autism. It was diagnosed at 1.5 years. He had a bunch of things that one might think of are likely mitochondrial, including that he got worse after vaccinations, he got hypotonia, low tone, he had OCD, and other things. And he had autism, but there were several things about him, chronic fatigue and other things that suggest that maybe that there's a mitochondrial component to his autism, as there is in many cases. Probably most cases of autism have a mitochondrial component to it. And then here's some of his issues. He has like constipation, fatigue, sleep problem, night twitches. All these things are functional disease things that are really common in mito. So we did the sequencing in him, and when you do the sequencing, particularly now that I'm doing whole genome sequencing, the number of variants that we find in each patient is in the hundreds of thousands. And because we each vary by hundreds of thousands of variants from other people. And to try to get that 100,000 variants to a small number that we can actually deal with, that's the real trick of the whole thing. It came up with 3 variants that I thought may or may not be related to his disease. The first one is the one that I thought was most likely related to his disease. This is a sodium channelopathy. We just discussed another patient that had a sodium 4A mutation. This is a sodium 2A mutation. They tend to have autism as well. And I was thinking I was going to tell them this is likely 2A disease. There's also a potassium channel, ABCC8, that causes some diabetic type problems as well, and I thought, well, maybe that could be it. And then my third one here was in the creatine transporter. It's the protein which moves creatine across membranes. It's necessary to get creatine into the muscles and into the brain. And it's a not uncommon cause for mental retardation and sometimes can cause autism. So when I went over with the family with these mutations, and this is actually cut and paste from the laboratory report. This is when I was at Cortigen. You can see that the creatine transporter, the laboratory had come up with these sort of things, and among them was aggressive behavior and autism. The parents were on the telephone when I was discussing this, and it was immediately, well, wait a minute, the creatine transporter really sounds perfect, and the other ones sound less likely to be it. The creatine transporter is on the X chromosome, so the mutation came from the mother, who's unaffected because she has a normal X chromosome. And based upon this, see, the patient had all of these. I don't know how easy it is to read that from where you are. But the point I'm trying to make here is that the variants need to be connected to the patient. When I see a new patient, I usually spend 3 hours in the room. Some of you probably were longer. And if it's a really easy case, it may be 2 hours. But I need to know everything about that patient, because when I look at the hundreds of thousands of variants, I need to know which ones are likely to be. And you see that the ones with the stars fit. That doesn't prove that that's what it is, but that makes it a candidate variant that may or may not be related to what he has. Now, he was tried on creatine, and creatine didn't work. Sometimes creatine works and sometimes it doesn't. If you can't get creatine into the brain because your transporter is destroyed, then giving creatine is not necessarily going to help. It's if the transporter works a little bit, then a whole lot of creatine might get enough of it into the brain. So then we tried cyclocreatine, which is something which is very difficult to find, but we were able to find it. It goes in the brain without the creatine transporter, and it made a dramatic difference. We used an OT that had been following Payton for many years and didn't tell him about it and asked him to see what the difference was. And these are the things that the OT said that were different when he was on cyclocreatine. So it wasn't like he was a little bit better. He really made a tremendous difference on that. And so this is not 100% proof or anything, but this is pretty good to me that the creatine transporter is involved in his disease enough so that getting creatine to his brain makes a difference. Yeah, he still has intellectual disability, he still has some degree of autism, because you can't reverse the last 18 years of his life and suddenly make him normal, but his behavior and his anxiety have improved dramatically, and his language output has improved as well. Okay. You all know your metabolic pathways, right? You notice that right here in the middle is the Krebs cycle. Did you see this? There's the Krebs cycle. Okay, right in the middle of the intake.
Oh my goodness.
Okay, so these are a simplified metabolic pathway. Where in the metabolic pathways have the cases that I've discussed with you so far? Where do you think they are in the slide?
Red box.
None of the patients that I've told you about are anywhere on this slide. None of them. Because for the mitochondria to work, you not only have the enzymes, but you have 1,000 proteins that are needed to regulate the enzymes, to make the enzymes, to put them in the right place, to tag them, the creatine transporter is not on here. You need creatine to run this stuff, but to get the creatine in there, that's just supply. If several of the mutations were channelopathies, they're not metabolic at all. What the mutation is it causes the energy to run out. They're not part of the metabolic pathways. The point is that even if you could sequence all of those genes, and that was the way that we did it 3 years ago, in the dark ages, you still won't find a lot of patients. You really need to sequence all of the genes. So, this is just a little thing I want to explain. I've said there's hundreds of thousands of variants. A very simplified way of looking at the variants is that variants can be rare. In fact, they can be novel. They've never been seen before. Or they can be common and they're present in a large percentage of people. The most common a variant can be is 50%, because if it's 51%, it is now the normal sequence, and the 49% is the variant, right? So variants can be from never seen before to 49.999% of the population. But variants can be small or large. And I'm going over this because when people say, well, these are the tests that we need, so you'll understand why one test won't do everything until now. Small variants are a single base pair. There's 3 billion base pairs in our genome. The genome is all the DNA. And then large could be millions of base pairs. Some patients have mutations which involve a deletion, that means you take it out, or a duplication, that means you add it in, of millions of base pairs. And they can be anywhere in between. So the first thing to talk about is the philosophy of medicine, is that Western medicine is trying to get a diagnosis, right? They need to put a little ICD-10 code on there. Once you have the diagnosis, then you know what the patient has, and then you know how to treat it. And if you don't have a diagnosis, that means that you either made up the disease or the patient's crazy, right? So that's kind of unfortunately the way things are. And there are casual variants that are very high risk. In other words, if you have that variant, you're very likely to have disease. There's no variant that says you have disease for sure. People thought there were, but even the variants which have been shown to cause disease, there are people in the normal population that have them. The variant may increase the chance of disease dramatically, but it doesn't cause the disease. It's a risk factor. So risk factors can be very low or they can be high. The more rare the mutation, the more likely it is to be a high risk, because obviously it's very common in the population. It's not going to almost cause the disease. The ones that cause the disease are going to be very rare. The ones that are common, at most, are risk factors. Most of them are normal. So the first things I want to get across is, yes, we do molecular technology, we do DNA sequencing to find the cause of disease. That's what everyone does. I do that too. But most people don't have it, because most of you in this room that I know of don't have a cause to your disease. They won't. I haven't found it, and I never will, because you don't have one. You have multiple factors. Each and every factor increases the chance of the disease. And that the disease is based upon many factors, not a single cause. And looking for the cause is not going to work. The whole idea of finding the diagnosis is really kind of falls apart. Most disease is polygenic. It's involved in multiple genes and also environmental risk factors. Diabetes, asthma, Alzheimer's, heart disease. These are all polygenic, multifactorial. They don't have a single cause. They have multiple causes. Most mitochondrial disease is polygenic and multifactorial. Most functional disease, most neurodevelopmental disease. Some of them are up here. Some of them we have a cause, but most of them we don't. Risk factor genes. A risk factor, so if it's not a de novo means that it's new and it's not in the parents. It was a new mutation in the child. Sometimes we find those, and I'll talk about those later, but most of these genetic changes came from the mother or from the father, about 50-50. So what about the parents? What about the brothers and sisters, the aunts and uncles, the grandparents? They have the same variant. If the variant is very high risk factor, then you have a situation where you can have a relative that has the same disease or a different disease, because remember in mitochondria that it causes energy metabolism problems, so you can get almost anything. Or you can have no disease at all and be a carrier, or you can have what's called a form frust, which is here. In a form frust, you have just a little bit. So someone with very severe migraine or cyclic vomiting may have just headaches. Somebody who has a form frust of autism would be like a billionaire, right? Because, I mean, what I tell my adolescent patients with autism is, name a billionaire. No, he's got autism. Name another one. No, he's got autism too. Because the same genes which predispose towards autism, what they do is they turn the brain inward. And a little bit of those genes are very effective in getting through school and higher education and being very creative. So it's oftentimes, not always, but oftentimes the parent of a patient with autism will be an engineer or a computer programmer or a scientist or an entrepreneur. For cyclic vomiting syndrome, often there are normal people that happen to have migraine or irritable bowel or something like that. So sometimes it can be that. But if it's a mild risk factor, it just increases the chance of disease a little bit. And the reason why the patient has it is it's got a lot of minor ones from the mother, a lot of minor ones from the father, and that one child happened to get a bad combination. Often the siblings will have lesser bad combinations and will have milder manifestations, or not at all. Okay, so if any of you had been in my talks like 5 or 10 years ago, you're wondering, well, I'm singing a completely different tune, because I used to say it all came from the mother, right? Back then, what I said is that maternal inheritance is from the mitochondrial DNA comes only from the mother, and the mitochondrial DNA is a very powerful risk factor for mitochondrial disease and any of the functional disorders and any of the neurodevelopmental disorders. So with this grandmother here is going to pass on her, what happened to this slide? That is so weird, it's completely normal on here. Anyway, this grandmother here is going to pass on her DNA to all of her children, including her daughters and her son. And her daughters are going to pass it on to all of their children, regardless who the father is. And her son's not going to pass it on to any of them because the mitochondria comes from the egg, not from the sperm. So everybody in red has the same mitochondrial DNA sequence, unless there's been a recent mutation. That is weird, it's the same thing. And this is a real family. You can see that there's different cyclic vomiting syndrome here, a lot of migraine, a lot of seizures, depression, irritable bowel, and other problems in the family. You can see that the people in black have more disease than the people that, and there's a lot of other people that should have that. But anyway, this slide's been, it's normal in here, but it's not on there. I hope that's not for the rest of the presentation. But anyway, this is what it really looks like, because if your mitochondrial DNA is not working quite perfectly, it predisposes towards many different disease manifestations. But why does everybody on this slide that has the same mitochondrial DNA, why do they have all completely different disease manifestations? Because of the nuclear DNA, because of the 23,000 genes which are on the chromosomes. And up until this point, I've been talking about those 23,000 genes on the chromosomes and how they operate. So it really is a combination of what is on the mitochondrial DNA and what is on the chromosomes. And so that's why you have to sequence both. You have to sequence all the DNA regardless where it is. Okay, so I've been using this word functional disease a lot. Most of you know what it is, some of you might not. There are many, many, many things that are functional. Functional is when the cells aren't functioning right, but their structures are normal. You can do an MR scan from head to toe and everything looks normal. All the structures of the cells, the tissues, the organ systems, they all look normal, but they're not functioning right. So you have pain, fatigue, GI dysmotility, things like bloating, nausea. You have dysautonomia, dizziness, palpitations. You have mood disorder, depression, anxiety, panic. These are things that you feel that are subjective, that you feel yourself and nobody else can really see. Nobody can tell if you have itching or pain or nausea or fatigue or depression. They are things that you feel yourself, and unfortunately, because they cannot be tested for in an objective lab test, they're often poo-pooed by physicians that don't know what to do with it or don't know if they're going to believe it. Functional disorders are defined by various types of doctors in different parts of the body. And here's some of the functional disorders. There are actually, like 50 or so in reality, but I put only some of them on this slide. The ones that I put in colors are the ones that I see a whole lot of in patients, but there's many different functional disorders. About a quarter of the population has a functional disorder. Functional disorders are by far the #1 cause of disability in our culture. Depression, chronic pain, chronic fatigue, anxiety, all of these things are by far the #1 cause of time off of school and time off of work, of disability. Cancer is #2 for disability. It's functional disease. But where is the government putting its money? Not in this area. I have to show the elephant, I always do. Is to show that different, yes, the elephant again. And it's lying down because of chronic fatigue. The same dirty, same rotten joke, right? Okay. So the idea is that this is a proverb from India, and that you have 3 different men, blind men, and they're filling parts of the same elephant, and they call it by different animals. And think about, so this is obviously your ENT, and here's your cardiologist, and this guy is the gastroenterologist. And they're all saying, one of them says tinnitus, or TMJ, and the other one says POTS, or psychological, or they all say that, and this one says irritable bowel, right? But it's the same patients have all of those. I've shown you over and over and over again, I've showed you patients that have many of the things on this elephant. And if you remember, some of them have mitochondrial problems, some of them have channelopathies, some of them have other things. They have many different things. They came to come together. I know that I've been spending a lot of time. Here's just another patient. This is her artwork, and she has lots of different things, and has a mutation in glutamate in this right here, which makes some glutamine to glutamate, which decreases the amount of alpha-ketoglutarate in the Krebs cycle. And then I put her on alpha-ketoglutarate, and this is the email that the mom sent, and she's gotten much better since we've increased the alpha-ketoglutarate many times, and she's gotten a lot better. So it's not, sometimes we can find an exact problem. This is in the energy metabolism. This is the first slide, this is the first patient that I've shown you that's on that slide, that blurred slide of the energy pathways. This one is on that slide. So sometimes they are. Same one I showed you before. I showed you last time that they can be rare or they can be common. But the other thing to say is they can be small or they can be large. Sequencing is actually reading the genetic code. In sequencing, you look for small mutations. In microarray, you look for large mutations. And if you could see that they do not overlap, there's a very, very tiny area in there that's getting smaller and smaller as technology gets better that neither will find. And there isn't any test out there that will find it, actually. We don't believe there's much in there, but who knows? So you really need, so it's a question of, if mutations can be large and mutations can be small, you really need to do both those technologies. Copy number variants. Copy number variants are another way of looking at large variants. You have too much or too little of something. You have a deletion or you have a duplication. They can be de novo, that they were not present in the parents, or they can be inherited from one of the parents. That's very important to know which one. If they're de novo and they weren't in either parent, usually it's the cause of disease, not always, and the recurrence risks are very low. If in siblings are not likely to have it. It's a new mutation. If it comes from one of the parents, then it's probably a risk factor, unless that parent is affected themselves, usually not. And so it doesn't cause the disease, it increases the chance of disease. But since a parent has it, and that parent will pass that copy number variant to half of his or her children, the recurrence risk for disease is quite high. This is just to show you that there are many. There are actually, in autism, as of 2016, were 478 distinct copy number variants that are associated with autism. There are so many now, I don't want to count them anymore. These are the more common ones. This is the number of published studies that have found that this copy number variant is associated with autism. So, if you have a copy number variant, the large mutations, they tend to have things like growth retard. You're small in size, you have intellectual disability, what used to be called mental retardation, it can include autism, or you have birth defects. Or your face looks a little abnormal, what you call dysmorphia, or small, tiny little birth defects to the face. Those are the common things, but as CMA technology has gotten better and better and better, the large mutations are getting smaller and smaller and smaller, so you find them in people that have normal intelligence, normal size, normal faces, no birth defects, they just have the disease. So when we used to do chromosome analysis, we wouldn't do it unless someone had birth defects or mental retardation or they were really, really small. But nowadays, the CMA technology has gotten much better. I use this to describe that the second most common of the copy number variants is associated with autism causes Angelman syndrome. And Angelman syndrome, they do have dysmorphic faces, but most people can't recognize it. It's not like Down syndrome, you can see it across the room. It's much more difficult to do it, and many expert physicians have missed cases, including myself before, because the faces is so normal looking in most cases. The problem with Angelman syndrome is that the copy number variation will not find all cases, and you do need to do methylation or imprinting to find it. So as there are some exceptions to the sequencing, will find everything under the sun. Sequencing maybe find 90%, but not everything. Okay, so that was looking at large mutations. Now we're going to go back to looking at small mutations, which we've been looking at for most of this talk. This is the machine. This is the Illumina machine. It's called a HiSeq that actually finds the mutations, and this is about the height. This is about the height of a man. So it's a fairly large machine. You heard about 20 years ago or so, they had the Human Genome Project, and then they sequenced a person, the entire, the 3 billion nucleotide for the first time, and it was a huge, big factor for out the entire world. Well, it cost billions of dollars and took scientists all over the world to do it. This machine will do it in an hour.
How much does it cost?
$1,800. Amazing, isn't it?
Yeah.
Okay, DNA testing. We used to do looking at little tiny parts of genes, then we went to sequencing the entire gene, and that was a big step forward. But an entire gene could be sequenced. It took a month at the laboratory and $5,000 to sequence a gene, but that was amazing. Then we went to small panels, like 10 genes or so, and then when I was at Cortigen, we did large panels. Instead of sequencing one gene for $5,000, we were able to sequence 1,000 genes for $10,000. So things were really doing better, right? A year ago, we were sequencing 23,000 genes, which is all of them, for $1,000. That's called exome. That's sequencing all 23,000 genes. And so you don't have to do panels anymore. Panels take a lot of time to make, and as soon as you make a panel, it's obsolete, because the next day another paper comes out. And you're constantly updating the panels, and you're only as good as your imagination, because a lot of patients don't have things on that. When you do all the genes, you find everything. It's not reliant upon that you put it on the panel, or you fought, or you got it right. You just sequence every gene. Of course, the exome or the genes are only 2% of all the DNA. The other 98% is the regulatory DNA. Now we do whole genome sequencing, which is all of it, all 3 billion nucleotides. And that is a lot of work, and the laboratory that I send to has just dropped the price down to $1,800 for that, for the actual sequencing. Okay, so
And why is it 27,000?
why do genetic testing? How am I doing? I'm way over like always, right? Why do genetic testing? Well, first of all, the right therapy. In some cases, the right therapy might be creatine, or cyclocreatine, or alpha-ketoglutarate. It isn't necessarily going to be a drug. And so you want to do genetic sequencing, because why try all these drugs if it's something over the counter that has almost no side effects will work better? The second one is the right drug. Sometimes the right therapy is acetazolamide or propranolol or Kytro. But I never would have tried any of those in those patients had I not had the DNA sequencing. And then the right dose. Like amitriptyline, a lot of you are on amitriptyline. Amitriptyline is a difficult drug to give. In some people, you give amitriptyline, they have horrible side effects, often because they don't break down the stuff. Oh, I'm not going to do it in 5 minutes. I'll try for 15. How about that? Okay. So, I mean, some people are slow metabolizers. In a slow metabolizer, you give a little bit of the drug and they break it down really, really slow, and it builds up and gets toxic. They have a lot of side effects. We can find that now when we do the sequencing. Some people are fast metabolizers. Their liver just eats it up. No matter how much you give, their liver's going to eat it up. So you may need huge doses. If they're slow or fast enough, you don't want to use that drug. But it tells you if that drug's going to be the right one to use and how much of the drug to use. So it's not just do you use the drug at all, but which drug and what dose. And of course, having a name and understanding what the disease is and knowing something about it, that can often be priceless. Okay. Do you sequence just the patient, or do you sequence the parents? So if you sequence all 3 of them, that's called a triome or a triome. Sequence just the patient, that's often called a singleton. The main reason to sequence the parents are de novo variants. It's a variant that's a new mutation that was found in the child that's not in either parent. There are other reasons to do that. If there's a family that has a lot of problems in it, you can find a variant that's in all of the people with the disease and not in the people without the disease. That can be very helpful. And the other thing is if it's autosomal recessive, if you have 2 copies, you can't tell if both copies come from one parent or one copy comes from each parent just by sequencing. You actually have to sequence the parents as well, and that can make a big difference. But the disadvantage of doing triomes is double the price. You have to sequence 3 people. It's not triple the price because the interpretation's not triple, but most labs double the price when you do a triome. So that's the main reason not to do it. And then there's sometimes both parents are not available, so then that can't be done. In reality, new mutations or de novo mutations are common in neurodevelopmental disorders. They're common in autism, and intellectual disability, epilepsy, and birth defects or / syndromes. So if you have intellectual disability, autism, epilepsy, syndrome, birth defects, then new mutations are common and a triome is really the way to go. If you don't have those, triome is better, but it's not absolutely necessary. So my testing recommendations for neurodevelopmental disorders. This is autism, this is intellectual disability, this is epilepsy, etc. Whole exome plus CMA for large mutations. So you do small mutations, you do here, whole exome sequencing, you do large mutations, you do mitochondrial DNA, as I mentioned. Fragile X is another one we didn't talk about. And then pharmacogenetic testing, that's where you see if the drug is going to break down fast or slow. Whole genome sequencing now does everything there, can do all of it. And since whole genome sequencing is cheaper than all of the above, plus you get a lot more information, that's what I'm going for now. And then the question is, triome, do you sequence the parents versus just the child? And then, Angelman syndrome, I mentioned methylation in some cases. And then the expert interpretation, that's the hard part because the laboratory does not know the child very well. They have a little bit of information. It's not the same as having followed the patient in your practice and having that. They can't go back to the family and ask questions. They're not necessarily experts in the disease that your child has. They're not going to spend a lot of time at it, and they're also very allopathic. And I think some of you know what that means. They're just straight and narrow. We can prove it causes disease, we talk about it. Otherwise, we don't talk about it at all. They're not really integrated in their technology or their outlook. So you really need to be able to get somebody that can interpret that. For functional disorders, cyclic vomiting syndrome, weird migraines, chronic fatigue syndrome, complex regional pain syndrome, things like that. It's, again, whole exome, CMA, mitochondrial DNA, and pharmacogenetic. It doesn't, the Fragile X been removed from that slide. I was doing all of this until a month ago when the prices were cut in half for whole genome. Now I'm starting to order whole genome on many of these cases just because you get all of it for cheaper, or about the same price.
Is the top one.
The top one, yeah. The top one is now about the same price as the next one down, so why not? And then the only difference is that in functional disease, singleton is usually good enough. We can trace it through the family once we figure out what the problem is. But you generally don't have to do a trio. Triome is still useful, and particularly if there's any neurodevelopmental disorders. But it's double the price if you're paying cash for it. If the insurance is paying for it, then yeah, get triome. Okay, costs. The costs have come down dramatically. We can do whole genome now for the same price. 10 years ago, we did a single gene. So, you know, the costs have come down, but they're still out of the range of some people, of many people still. We can do an exome for as low as $1,000. That's the sequencing cost. That's not the interpretation cost necessarily. Mitochondrial DNA and CMA are $500 each. You can see it'll be $2,000 if you do all three of them, and then a genome includes all three of them plus more, so it's even a little cheaper. So that's why I'm saying genome now. Even a month ago, I was getting these because this was $3,000 or $3,600. Some laboratories take insurance, some are cash only. Some laboratories take insurance, but the cash prices are really horrible. So if you go to that laboratory and the insurance doesn't pay for it, then you have to go to another laboratory that has a cheap test for cash. I like the laboratories that will take insurance, and if the insurance doesn't go through, they have a cheap price. So you go there, you get it. And there are some companies that will do that. The ones I'm using today, which may not be the ones I recommend next week, because everything is, I remember free agent. For exome, there's a company in Germany called Centogene, which is really, really good. Those are the prices I've shown you up there. And then there's an Israeli company called Variance. They're the ones that came down to the 1,800. I've gotten a few genomes from them, and they have great interpretation and everything. You can get cheaper prices for a genome, but but there's no interpretation. It just gives you a sequence, and it's not helpful.
Have you heard of Gen X?
Gene DX, or is it?
Yeah, maybe that's who it is. They have ours right now, and they've had it since February. They tried to bill our insurance $27,000. It keeps getting denied, and now they're wanting to work out a price with me. Their sequencing is fine, but their interpretation lacks imagination. We're going to say that. Okay, so now they're wanting to work something out if I want to get.
There are much cheaper alternatives out there. You can get for exome or genome.
They did exome, and they did it for me, him, and my parents.
A trio
of exome. Dad, so they did it on 4. They did my mom, my dad, me, and him.
Yeah.
So
yeah, I know. Centogene will do an exome in the 4 of you for $3,000. And they will run it through insurance, and if it doesn't
no,
go...
not your dad, because we didn't have your dad available. Your dad's cooperative.
Okay, so...
no.
So it's much more likely to get the insurance to pay for it if you have a letter of necessity and if you use the appropriate codes. I mean, for instance, if you have epilepsy and autism, you put epilepsy as the top code and autism as the second code because they're more likely to pay for it for epilepsy. But that's the kind of thing that a lot of people don't know about. But interpretation is the thing. The interpretation costs, they're my costs. What I do is I go through the entire sequence. I charge $850 for exome and $1,000 for genome. And insurance doesn't generally pay for that. What I do is I look at every single variant. This is a patient with autism. Some of you might have heard Temple Grandin, Dr. Temple Grandin. Yeah, this is her sequence.
Wow.
So we're publishing this together. This is alphabetical order of all of the genes. And then these are the variants in the genes, and then the various different computer programs around the world. It actually, what it does is it takes the data and it throws it on the internet to many computer programs all over the world, gives me numbers, and then color codes it. You can see there's a lot of green on here. Green is like, it doesn't look like it's related to disease. And then the sequences that I thought in Dr. Grandin that are most likely to be related to disease, I put on this one here. You can see there's a lot more red than there was over here. And in addition to finding several variants that likely underlie her autism, I also found the reasons why she gets, why she has these episodes in which her feet suddenly get really hot, and then her body gets really hot, and she has to run out of the room, and then she has these episodes of her fever and other things like that she gets that are really disabling for her. Mutations cause that exactly that we found on there. Plus, I found out why she's missing 6 teeth and many other things. So,
Wow.
she was very excited to learn all this information on that.
But
So.
in something like that, you found all these causes, how many of them are actually treatable in some way?
Well, the thing that, I mean, she goes around the country, talking about autism. To her, autism's a net positive.
Right.
Right, for her,
Having
but,
her feet
but,
get
what,
all hot.
but what bothers her are these episodes in which she feels really hot, and then she gets palpitations, and she gets short of breath, and all of this stuff. And then she has whole body pain. That, I found that the mutations are in the MEF gene, and that's treatable.
Wow.
Which is wonderful.
Yeah.
Okay, wow. So that's you.
Okay, so a lot of the stuff you're talking about, functional disease, you're talking about pain and fatigue and GI problems and autism and stuff. Unfortunately, sometimes when the medical team looks at you, is this what you think they see sometimes? I've been talking about it throughout the talk. This is very complicated, and it's a different way of looking at medicine. And unfortunately, when your child is getting sick with weird stuff that no one can figure out, and most of it's functional, and all the tests are normal, and things come and things go, and then new things come and then they go, and then new things come, and the child has lines and everything. They start thinking that either mother's doing something, that mutation, like medical child abuse, factitious disorder, Munchausen, or they start to think that this is psychiatric, conversion, somatization, or just caused by stress if they're being nice. Unfortunately, these cases have increased throughout the country. I've been a witness, an expert witness in, I think, 5 states. I have cases currently going that I'm an expert witness in, and many of my patients have also been accused. And I gave a talk to Mito Action a few months ago. It's still on the website, right? Okay, and it goes over this much more in detail, and this slide, I think, is one of the slides that's on there. But it goes over things, how can you avoid being reported? The things to do differently, and still be who you are, and still advocate for your child, but just not to do those things that really, really upset the doctors and make them report. And then what to do after there's an allegation. And this really all boils down to having a physician that goes to bat with you and says, no, this is real, and this child needs to be treated, and this mother is being appropriate. And if you file, I will testify against you. That's what really helps. Okay, Caroline, can you make a few samples? This is my helper. So, molecular data makes a huge difference. Treatment is dependent upon the individual. There is no magic formula that will help everybody. And it needs to be individualized. For example, if you have migraine, you need a lot of riboflavin. If you have constipation, you need a lot of magnesium, etc. And by the genotype, if you have certain genotype, you need alpha-ketoglutarate, or you need creatine, or whatever. But there are certain things that everybody with mitochondrial dysfunction should have that are helpful across the board and have 0 to very, very few side effects. And everybody was getting these. Coenzyme Q10, carnitine, riboflavin, magnesium, all of these things that everybody was getting, and the average patient was getting 10 different supplements. The largest number I found was 40 in one patient.
Wow.
The average was 10. And it's crazy to get 10 different supplements from like 5 or 8 different companies. Not only is it expensive and very difficult, but you have to get the kid to take it all, and you have to prepare it and everything. So people kept on bothering me to do this, but to actually make my own product is a huge amount of work. And then Cortigen went out of business, and I suddenly found myself with extra time. So that's what happened. So I took the time and I made this product, and it's really what I've been doing. I had spoken to all of these doctors and many more, and these are the people that are involved in that. What do you use in your patients? What causes side effects? What doses are safe? What do you recommend? What brand of this and everything? And we put this together. Spectrum needs, it says Spectrum like for autism, but I use it for cyclic vomiting, I use it for mitochondrial dysfunction, I use it in chronic fatigue.
Can you read the doctor's names, please?
Oh, yeah, this is Dr. Corbier, he's in South Carolina. Dr. Ellis, New York City. This is Dr. Fry. Some of you might know him. He's a big mito guy. He's now in Phoenix. He was in Arkansas. Dr. Rosignol, he's an autism doctor from Florida, and he's now also in Orange County. Dr. Mintz has a neurological practice in New Jersey. Dr. Skolron is a naturopath from New Jersey. And then Audrey. I always know her as Audrey with the grease spot. She's from West LA. She's a developmental specialist. And there's been many others that I have gotten involved in doing that as well. And we're coming up with a second product pretty soon called Mito Needs, which will be the same thing but with a higher CoQ, a little bit more vitamin D, a little bit more carnitine and everything. But basically what I've been doing to make Spectrum Needs fit for mitochondrial patients is we use a lot of extra CoQ, and then of course each patient's individualized to what they need on top of that. So what I'm doing now is I'm having my patients on this plus CoQ. It's a powder form. Some of you are on it, some of you are not. You can taste it. It's in water, if anyone wants to taste it. And then I have some here that I can sell. Normally it sells for $77 on the internet, and then it's $85 once you pay for shipping. I can sell them for $70 here if anyone wants some. The other thing is in interpretation. I mentioned that the interpretation is what matters, but not everyone can come to California. You're all here, so it's probably not as relevant, but for patients across the country that want to have DNA testing, I mean, this is going to be on the internet, so some people may be in states, much further away. What they can do is that I can help your doctor order the appropriate tests. I can get my discounts, because these labs that I have these prices for are highly discounted prices. So even if you add my interpretation in, sometimes you save money because the discount can be greater than my interpretation fees in some cases. And then I can interpret it, and then I can discuss it with your physician. If you are in a state physically in which I have a medical license, you can be on the phone while I discuss it with your physician, and I can talk to you about it. I currently have medical license in California, New Jersey, Pennsylvania, South Carolina, and Florida, and in the process of getting it in Arizona. It's extremely expensive and takes a lot of time to get medical license. Getting it in 50 states is just not practical. Keeping up with the 50 states requirements would be taking a full-time job. It's hard enough with 5. So this is an option. This goes through New Jersey. Dr. Mintz was one of the ones that you saw in the Spectrum Needs. He runs the neurological place called CNNH in New Jersey. And it's, people can be anywhere in the country, and we're going international with this as well. And I have business card. If anyone's interested in this, you can get the business card for this practice. It's right over there. I also have NeuroNeeds business cards if anyone's interested. And then here, I'll leave this up. This is going to be on the internet, so any of this on there you could be interested in, but this is the information to get the CNNH neurogenomic program I just spoke about, my private practice, neuro needs. The information that I've discussed about genetic testing is in even more detail on my website, if anyone's interested in getting the larger version, of which right now I gave you the very short version. And I'll just leave this up there, and if there's I think that we'll turn off the tape now, right? So goodbye.